Okay, guys, so in this lecture, we are going to talk about the structure of the heme group. Okay, what is a heme and what's its function in different proteins which are present. Okay, heme is basically an organic molecule, and it is, I should rather say, it is a complex ion or a complex structure which is formed between a ligand molecule and a centrally placed iron ion, basically a ferrous ion. When both of these coordinate with one another, they form the heme structure, right? And the function of heme structure varies from protein to protein, and it depends upon the type of protein in which it is present. It depends upon the arrangement or the micro environment around the heme group, which is created by the protein chains. uh, the arrangement of the globin chain dictates the role of heme group in those proteins. Okay, so it basically contains a tetrapyrrole ring, which is also which is the type of tetrapyrrole ring is basically the protoporphyrin 9, which is present within the heme group, and a centrally placed ferrous ion. Okay, what is a tetrapyrrole ring? We're gonna talk about that now. Okay, tetrapyrrole. First of all, let me tell you the structure of a pyrrole ring. Okay, pyrrole ring, this is a cyclic structure which contains a nitrogen atom and four carbon atoms, and there are alternating double single bonds. If we take four of these rings and join them together through some bonds, through basically methylene linkages, these are the bonds which are present between subsequent pyrrole rings, and we basically do this for four different pyrrole rings forming a closed cyclic structure. Then that structure which forms is a tetrapyrrole ring. Okay, and I'll show you a diagram over here. This is taken from your book Lippincott. Okay, and over here, if you look at this structure, this ring over here, the nitrogen is double bonded to a carbon, which is single bonded to another carbon, double bonded to a carbon, then single bonded to carbon and nitrogen. This ring is your pyrrole ring. This is the first pyrrole ring, and this first pyrrole ring is bonded to this pyrrole ring is bonded to another pyrrole ring through a methylene linkage. Okay, this is your methylene linkage. Basically, this entire structure. Okay, methylene linkage. Why is it called methylene? Because it contains meth means one, and it contains only one carbon atom in this linkage. So this is a methylene linkage present between subsequent pyrrole rings. So this is a second pyrrole ring, then there is another methylene linkage, then a third pyrrole ring, another methylene linkage, and a fourth pyrrole ring. So these are basically the four pyrrole rings present in the tetrapyrrole ring structure. A type of tetrapyrrole which is present over here is known as the protoporphyrin 9, and it basically contains a few branches as well. This is a propionate branch, or if you want to name it using the IUPAC naming, then it's propanoate. Okay, propanoate or propionate, three carbon acid. Then there is a methyl ring as well, methyl branch, sorry, not a ring, branch. And then we have a vinyl branch as well, vinyl branch. And yeah, these are the three types of branches which are present on the pyrrole rings of a protoporphyrin 9. And this protoporphyrin 9 is present in hemoglobin and myoglobin proteins, okay, which are basically the most abundant sources of heme groups in the body. Hemoglobin has a role in the transport of oxygen, as you guys already know, and myoglobin has a role in both the storage and the transport of oxygen. Okay, myoglobin basically it transports it. Its primary role is to store oxygen, but it actually transports the oxygen when the partial pressure of oxygen within the muscle cells becomes very low. So it then supplies the oxygen to the muscle cells as well. When oxygen demand increases, so myoglobin also supplies the oxygen to the muscle cells. Anyways, coming back to the protoporphyrin 9 structure, uh, these the nitrogen atoms, they use their lone pairs to form coordinate bonds or dative bonds with the centrally placed ferrous ion, and this ferrous ion forms four bonds with a tetrapyrrole ring, but it can form a maximum of six bonds. Okay, so two of the bonds which are right perpendicular to this ring, they are free. Okay, one is above and other is below the ring. Okay, these two rings bonds are free, and these bonds, one of them is then used to bind with the molecular oxygen that it has to transport in myoglobin, in hemoglobin and myoglobin, in which it has to store, and the other is bound to a histidine amino acid, and we are going to talk about this in detail when we are going to look at the structure of myoglobin in the next lecture, and the link to that lecture is given in the description. Do check that out. Right, so this is basically the tetrapyrrole ring. This diagram is taken from your book. Lippincott and iron can form six bonds, four with porphyrin nitrogens, and two additional bonds, one above and one below the planar porphyrin ring. Okay, this porphyrin ring is planar. Okay, as you guys can see, each of the carbon atom is has a planar shape in here. Each of the carbon atom has a double bond and two single bonds. This is if you look at it using your valence shell electron pair repulsion theory as well from your chemistry, then as well you can see that each of the carbon atom is planar. Okay, so therefore this entire ring is a planar ring, and right perpendicular to the ring above and below the ring we have two bonds which are free. One of them binds to oxygen, molecular oxygen, and other binds to a histidine residue. Then I've gathered some information over here. Let's read it out. One of the two free bonds bind to the molecular oxygen and other bond binds to histidine residue. All right. And I told you that I'll talk about this in the hemoglobin and myoglobin structure as well. Okay. So one of them binds with molecular oxygen. It's this free bond that is responsible for the transport of oxygen in hemoglobin. Ferrous ion can form a maximum of six bonds. Four of them are already bonded and two are free. The free bonds are perpendicular to the protoporphyrin 9 plane. Right, so we've talked about this entire structure as well. Right, so now the proteins which contain the heme group, they are called heme proteins. Okay, those proteins which contain the heme groups are called heme proteins. And basically there are some special proteins which contain these heme groups, and those proteins which contain the heme group as permanently attached or permanently bonded to them, the heme group in those proteins, if is permanently bonded to them, is called a prostatic group. Right, so this is something you guys should know. Prostatic group is also present in enzymes, and in enzymes you guys know that the coenzyme which is permanently bonded to the apoenzyme is known as a prostatic group as well. Okay, this is an organic molecule. Prostatic group is covalently bound non-protein organic molecule. Right, it is not the protein part, it's the globin part that is the protein part in hemoglobin and myoglobin, but the non-protein part is the heme group, which is actually the prostatic group of that hemoglobin or myoglobin, and it's permanently attached to the protein part. And prostatic group, as I told you, are also present in enzymes. A function of the heme group, it depends upon the type of environment in which it is placed. Okay, a type of environment which is created by the globin part of the protein. Okay, it is present in a variety of proteins, and as I told you, those proteins are called the heme proteins. In cytochromes are the electron carriers in the electron transport chain in mitochondria, in the cristae of the mitochondria, and they are components of the electron transport chain. They accept electrons and then they transfer electrons, and within the cytochromes it's basically the heme groups that are actually performing the role of accepting electrons. Okay, they accept electrons. Then in this enzyme called catalase, or even in peroxidases and catalases, heme groups are present as a constituent of the active site. And heme group as a constituent of active site, the role of catalase is degradation of hydrogen peroxide when it is produced, because since you guys know that hydrogen peroxide is toxic to the cells, so it has to be broken down by catalase. And so in catalase, In catalase, the arrangement of the protein part is such that it makes or it dictates the heme group to function in the active site for the degradation of hydrogen peroxide. Okay, so in catalase, the prostatic group is the heme group. All right, and finally, we talk about myoglobin and hemoglobin. These are the most abundant sources of heme in the body, and their globin chains are arranged in such a manner that they dictate or they cause the heme group to reversibly bind with oxygen. Okay, so there, and we are going to talk about the structure and function of myoglobin and hemoglobin in great detail in the upcoming lectures. And I'll give the link to those lectures in the description. Do check them out. And thank you so much.